So, author Jerry Kint once said, Alzheimer's is the cleverest thief because she not only steals from you, but she steals the very thing you need to remember what's been stolen. After hearing about my friend's difficult experience of witnessing his grandfather struggle with Alzheimer's, or AD, I was motivated to learn more about this disease and help ease the journey of having it. Hello, my name is Sruthi Potluri, and I am a high school senior from New Jersey. I am completing my research project under the guidance of my mentor, Andrew Bender. My love for genetics and neuroscience, as well as my desire to help individuals and families like my friends suffering from AD, led me to research the genetic factors associated with this disease. I also want to learn to apply computer programming to biomedical research. I decided to conduct a dry lab project where I worked on creating a machine learning model that predicts potential genetic factors of Alzheimer's. While there is a lot that we don't know about this disease, research has found that that Alzheimer's is marked by mutations within the tau and amyloid proteins. The tau protein is located in brain neurons, which we can see here, and buildup of the protein, also known as neurofibrillary tangles, is commonly found within individuals with AD as it leads to memory loss. The amyloid protein is located in many of the body's organ systems, and malformed versions of this protein, called amyloid plaques, are found between neurons of individuals with AD. So that's what this diagram shows here, causing neurodegeneration. Through my project, I hope to learn more about these proteins by finding other factors related to them in AD. The specific questions I want to answer are, how do genetic factors differ for normal aging and AD? What do they indicate about cognitive decline associated with AD? And how can gene expression be used to identify individuals at high risk of AD? My mentor and I used RNA sequencing data from the Allen Institute of Aging, Dementia, and TBI study, which contains 377 examples of gene expression levels, importing it into a Google Colab notebook to create our model. Using scikit-learn, we analyzed all the different files of the dataset to build a final data frame with all the information needed for the computer to detect patterns from the original data and create a model that's based on logistic regression. We later tested the accuracy of our model, making changes to the model as needed. The technique we primarily used was cross-validation, where our model set aside some data for testing while used the rest for training. Cross-validation enabled us to understand how well our model could generalize to untrained data. So in this diagram here, we can see how some data is set aside for testing and then the rest is used for training in each iteration. And the model keeps on doing this through each iteration until it finishes testing all portions of the entire data set. So we have successfully finished building our model, which has an average accuracy of approximately 84%. Now I am working on analyzing the results of our model, and I'm writing a research paper on them. By using values known as beta weights that are derived through logistic regression, I am able to identify which of the expressed genes that the model analyzed are most upregulated and downregulated in AD. The one protein that our model shows to be a strong indicator of AD is a glial fibrillary acidic protein, or GFAP gene. This gene codes for the glial fibrillary acidic protein, which is a pretty creative name, and it helps provide strength and support to cells and has been found to be linked to AD since it is correlated with amyloid plaque density. As I continue my analysis, I expect to find more proteins that are related to amyloid in tau and use my findings to help diagnose and identify potential drug treatments. I hope to contribute to advancing care for Alzheimer's as we seek to return the time and memories that have been stolen by the cleverest thief. Thank you.
All right, thank you so much, Sruthi. Great presentation. Any questions about her presentation? I have a question regarding, I asked this to everyone, but what is your inspiration? Is Alzheimer's something that's affected your family or someone personal to you?
Yeah, so it's actually affected my friend. He had a grandfather who was suffering from Alzheimer's, and he was very close with him. So hearing about like how he was always like trying to find ways to progress to treatment for Alzheimer's, whether it was starting a fundraiser, was just
Mhm.
inspiring for me. And I also wanted to try to help him and like other families struggling with this disease. So that's why I decided to focus on this disease as my topic.
Got you. Excellent. And if you had to take this a step further, if you had the opportunity to keep working on it, what would you do at this stage?
So there would be two main things I would do. So the first would be improving the model that my mentor and I worked on. So we used the logistic regression as a foundation for our model. So that's like a statistical method. There's other there are other statistical methods that are more complex and that would also do a better job of analyzing the data. So currently our model's accuracy is 84%. And if we were to use more advanced analytical methods like analytical like foundations for like the model, then we would be able to reach a higher accuracy. So I would try to explore that. And the other thing I would do is just trying to spread awareness about the disease and like
Mhm.
try to tell people like how like and try to just present the different proteins that my model has identified.
Mhm. In your research, did you look into some of the causes of it, and what are some things people can do to prevent the onset of Alzheimer's?
So I didn't look into how you can prevent it. I did look into the cause the cause. So the cause is also not it's not like clearly known. We just know that it's caused by some mutations within like the amyloid and tau proteins, but that's something people are still trying to research. So I
Mhm.
think that's also another reason why I was drawn to this exploring this disease. There's so much about it that we don't know yet. And it's
Mhm.
such like it's a disease that affects like such a huge population. So that's just something that I would want to continue looking into.
Yeah, there's like a lot of opportunity for research because it's such a big area of unknown. Totally.
Exactly.
Thank you. Any other questions from the audience or panelists? Doesn't look like it. Oh, Alex, you do have a question. Let me call on you. Go ahead. You should be able to speak now.
Sorry, I accidentally pushed it. Did not have a question.
Oh, you didn't have a question? Okay. So let's keep moving on then. Thank you, Sruthi.
Thank you.